The first thing that I want to talk about today in genetics video 5 is the difference between pleiotropy and what we call polygenic traits. So first, pleiotropy is a situation that we have when a single gene influences multiple phenotypes. So think back to earlier videos. We talked about maybe you have a gene that we denote gene B, and gene B just affects your eye color. Well, in pleiotropy, we would have one gene, so just gene B, but it's influencing more than one trait. So an example is the gene for sickle cell anemia. And if you're homozygous for this gene, there are many different phenotypes you see. You see abnormal sickle-celled blood cells, you see the breakdown of red blood cells, the clumping of cells, kidney failure, all these things. So you're seeing one gene giving you many, many phenotypes. Kind of the opposite situation is where you have many genes dictating one phenotype. So many genes determining just one phenotype. So this is the opposite of pleiotropy. And this is called polygenic traits. And if you look at the root to this word, this kind of makes sense. Poly means many, and genic means genes. So we have many genes determining one phenotype. So one trait that behaves this way is actually eye color. So in humans, there is not just one gene for eye color. There's actually, we know of at least 2. And maybe there's even more. There could be even 3 or 4. We're not really sure. And so instead of denoting eye color as just using one letter, because it's controlled by 2 genes, we actually use more than one letter. So if you were recessive for both gene A and gene B, you would have very, very light eyes, like blue eyes. And if you had 4 dominant alleles for the 2 genes, you would have very, very dark eyes. So some other examples that we know of that work this way, that are polygenic, are things like skin color and height. So let's look at an example. So here I have a Punnett square for eye color. So I have a mother here who has kind of a medium brown eye color, and her genotype is capital A, lowercase a, capital B, lowercase b. So the 4 different gametes she could produce, the 4 different eggs she could produce would be 2 capitals, a capital, a lowercase and a capital, and 2 lowercase. Similarly, the father has the same genotype, so he could make the same 4 sperm as the 4 eggs. And if we do this Punnett square, we see wherein the offspring gets all dominant alleles, they have very, very dark eyes. When they have all recessive alleles, they have very, very light eyes. And in between, depending on what alleles you have that are dominant and which are recessive, it would give you either darker or lighter eyes. Another example of a really well-known polygenic trait is skin color. And we know that skin color is controlled by at least 3 genes, so the A, B, and C gene. You can see that written there. So if you have 6 recessive letters, you kind of have the lightest skin. And if you have 6 dominant letters for the 3 genes, you would have very, very dark skin. So if I cross 2 heterozygotes, I would see that the offspring, I would get this huge variation of offspring depending on how many dominant and recessive alleles they had. And this huge variation, what we call a spectrum, is kind of a hallmark of a polygenic trait. You know a trait is polygenic if you get many, many phenotypes out, and they fall between 2 extremes, and you get the spectrum. So here, if I'm looking at this picture of skin color here, I have some children who have very, very light skin, and some who have very, very dark skin, and all the phenotypes in the middle fall along this gradient, this spectrum. So if you see a spectrum, you know we're talking about a polygenic trait. So obviously, if we're dealing with something like skin color, and I had a question like, okay, I have 2 parents with the following genotypes. So for skin color, this is the genotype for one parent, and this is the genotype of a second parent. And I say, okay, I want you to do out a Punnett square and calculate what's the probability of having a child with the darkest possible skin, all capital letters. And you might be thinking, well, how in the world do I make a Punnett square for that? And the answer is you don't. That would be ridiculous. That Punnett square would have a ton of squares, and it would be a waste of your time. So if you remember, earlier We talked about using probability to solve these kind of problems, and that's exactly what we're going to do now. At this point, the problems are too complicated to use Punnett squares, and instead, we could use the laws of probability, and it'll be much simpler to solve these problems. So this is the same problem here that I showed you on the previous page. So these are the two parents, and these are their genotypes for skin color, and I want to know what's the probability of getting an offspring all dominant for skin color, so the darkest possible skin. And the way you do this is you look at each gene separately. So first, we're just going to look at the A gene. So right now, you can kind of think of the problem as, if I had a parent who was heterozygous for gene A, and a parent who was homozygous dominant for gene A, what would be the probability of me getting a child who is homozygous dominant for gene A? So I'm going to shrink this. So I have one parent who's capital A, lowercase a, and the other parent is capital A, and I want to know the probability of the child being capital A, capital A. Now you could draw out a Punnett square here. So if I did that, I would see that the probability of getting a child who is capital A is 2 out of 4, or 1/2. The other thing you could do is use your rules of probability. So let me get my eraser here. So I know this parent here has a 1/2 chance of donating a big A, which is what I want if my child's going to be big A. And this parent has a 1 out of 1 chance of donating a big A. So because these are independent events, I can multiply them together, and I can say that the probability of my child being big A is 1/2. Okay, so let's go back to our problem now. So the probability of just getting big A, you can say is 1/2. So now look at just the B gene. So if I had a heterozygous for B and a heterozygous for B, the probability of getting capital B, if I do up my Punnett square, I see is just 1/4. So the probability of capital B is 1/4. And then the probability of getting capital C, if I have parents who are heterozygous and homozygous dominant, if I do add a Punnett square, we'll see that that's also 1/2. So now, because we're going to assume that these three genes are on different chromosomes, the gene A, gene B, and gene C act independently. So I could actually use my rule of multiplication here and multiply my three probabilities together, and 1/2 times 1/4 times 1/2 gives us a chance of 1/16. So that means the probability of having this offspring from those two parents is 1 1/16. So let's do another example. So what would be the probability of getting this genotype given the same parents? Well, let me erase a little bit here. I've made a mess. Okay. So let's go one gene at a time. So the probability of getting just a capital A from those two parents. This parent has a 1/2 chance of donating a capital A, and this one is always going to donate a capital A. So my probability there is 1/2 for capital A. If I cross two heterozygotes for B, I did that Punnett square over here, you could see the probability of getting a heterozygote is 2 out of 4, or 1/2. And then the chance of getting capital C, lowercase c, with these two parents, if I do that Punnett square out, and the letter C is awful for Punnett squares because the capital looks like the lowercase, we'll see that the chance, again, is 2 out of 4, or 1/2. So So then finally, these genes are all acting independently, so we could use our rule of multiplication and say 1/2 times 1/2 times 1/2 gives us a 1/8 probability chance of getting this offspring from those two parents. So now we're going to switch gears a little bit and talk about a different topic, unrelated to polygenic and pleiotropy, and that is linked genes. So at this point, we have been assuming if I had an individual with this genotype, I assumed that the A gene was on a different chromosome than the B gene. So if I was going to draw out their chromosomes, I would assume that I have one homologous pair with the A gene, and then a second homologous pair carrying my B gene, which means that this individual, when they underwent meiosis, they could donate a capital A and a capital B together, like these two chromosomes together. They could donate a capital A and a lowercase b, a lowercase a and a capital B, or a lowercase and a lowercase. Because the genes grow on different chromosomes, they can be inherited independently. However, when we're talking about linked genes, we're talking about two genes that are actually on the same chromosome. So what this would look like would be one homologous pair where the A gene is on 1/2 of the chromosome and the B gene is on the other half of that chromosome. So it's kind of like the A and B are physically linked together. They're physically stuck together. So if we look at an example in flowers, we could say that flower color, which we're going to designate as gene P, and pollen shape, which we're going to designate as gene L, are on the same chromosome. So capital B is going to be purple flowers, lowercase p is going to be red flowers, capital L will be long pollen, and lowercase l will be a round pollen shape. So if we look at this example, if I crossed two parents, or if I had a parent where it's telling me that on one chromosome, capital P is linked with capital L, and little p, lowercase p, is linked with little l, and it wants to know, well, what gametes can this parent make when they undergo meiosis if P and L were on different chromosomes, which they're not. You would expect, okay, capital P could go with capital L, capital P could go with lowercase l, little p could go with capital L, or little p can go with little l, if we had independent assortment. But because they're linked, because they're stuck together, the capital P can only go with capital L. So that means the only two gametes this parent could make are capital P, capital L, or little p, little l, because they're physically stuck together. So they can't sort independently. If you notice this word mostly here, the only exception, we can only mix them up. We could only get capital P with little l or capital L with little p if we did crossing over. So they're mostly like this, and a little bit of the other combination if we get crossing over. So you can kind of see here a picture. If this is a homologous pair, so both these chromosomes are like chromosome 1, for example, and if this cell were going to undergo meiosis, this capital P here is stuck with this capital L. So one type of egg or sperm they could make is capital, capital, and then the little p is stuck with the little l. So the other egg or sperm they can make are little p, little l. And the only way we can unlink them is if we do crossing over. So remember, during meiosis 1, prophase 1, homologs undergo crossing over. And crossing over is the only way we can get a different combination of alleles. So it's kind of like now we're adding in another layer of complication. When we're looking at a genetics problem, we have to know if the genes are unlinked or linked. If they're unlinked, they can make 4 possible gametes, but if they're linked, they're going to make 2 possible gametes most of the time. Okay, So just as a reminder, the small fraction of offspring with the recombinant phenotype. So that means the different combination of letters than the parent. In this case, it'd be capital P with little l, or capital L with little p. You only get that different combination due to crossing over. So that's it for video 5. Thanks for watching.